So welcome to today's education session, the last one of our National MedLab Week celebrations at UHN, sponsored by Biorad. Today we will have talks from two of our newest staff members in Core Lab, Sidney Pang and Thuy Huang. Sidney did his undergraduate at the University of Toronto in molecular biology and immunology before going on to attend the Michener Institute for Medical Laboratory Science. He did his placements at UHN as well as Mount Sinai for microbiology. His position in the Core Lab is his first professional role. Sidney will discuss the 3-week rotation he experienced as a student in the mass spectrometry lab. Thuy completed her advanced diploma for medical laboratory science at the Michener Institute of Education at UHN in February 2019 after completing her biological science degree with a minor in brain cognition at the University of Guelph. She completed her clinical placement from September 2018 to February 2019 at Toronto General Hospital and Mount Sinai for microbiology. Thuy will take us through her experience validating the EPOC blood system analysis for the point-of-care team that was to be used in the operating room. She selected this project as part of an enrichment rotation offered during her clinical placement. Without further ado, here's Thuy with her presentation.
Good afternoon, everyone. Thank you all for attending. So, as Laura mentioned, my presentation will be on the evaluation of blood gases on the EPOC blood analysis system. So I will start my presentation by briefly going over what point-of-care testing is. Point-of-care testing is a type of laboratory test that is completed outside of the laboratory at the patient's bedside by non-laboratory personnel. This can include nurses and respiratory therapists. One benefit of using point-of-care testing is that it reduces the turnaround time by delivering immediate results right at the patient's bedside. Doctors can then make immediate medical decisions based on these results, and this can have a positive impact on patient care. Rural communities can also benefit from point-of-care testing as they may not have access to large analyzers in their hospital. Point-of-care testing instruments can eliminate the need to send the samples out to larger hospitals and thus having to wait for the results. So, EPOC blood analysis system is a type of point-of-care instrument. It is a handheld cartridge-based system which measures blood gases, electrolytes, and metabolites. How it works is that the BGM test card is inserted into the reader component of the EPOC. The card takes about 165 seconds to calibrate, after which you have approximately 450 seconds to introduce the blood samples using a syringe. It needs approximately 100 microliters of blood to complete the test. Internal QC checks are also performed on each card. If any QC fails for any of the parameters, the result for that particular parameter would not be shown on the screen or the host of the EPOC. This is to prevent erroneous or invalid results from accidentally being used or released. After the blood sample is introduced into the card, the test takes about 30 seconds to complete. So, for my project, I was involved in validating one of the 2 EPOC instruments that the point-of-care team has received to be used in the operating room. The parameters to be validated include pH, partial pressure of oxygen and carbon dioxide, sodium, potassium, chloride, ionized calcium, lactate, creatinine, and hematocrit. So the first thing I have to do before I can start my project is to set up the EPOC, and this includes entering the correct units, reference and critical ranges in the instrument, making sure the printer configuration is correct, as well as enabling the tests or parameters to be used. After which, I also have to learn how to use the instrument. So that can include learning how to handle the test card properly, inserting the test card into the reader component, and as well as introducing the blood sample into the card. It takes a little bit of technique to learn and to get used to, but it wasn't too hard. After that, I was able to start my validation project, and it is divided into 3 parts: the linearity study, precision study, and method comparison study. So for the linearity study, there are 5 levels of blood gas and 5 levels of hematocrit calibration verification materials that were to be ran in duplicates onto the EPOC instrument. For the precision study, I were to run 3 levels of gas ISE control materials and 2 levels of hematocrit control material and duplicates as well for 5 days. It took me about just over a week to collect all the results between the 2 studies, and I was able to transfer those results onto the Excel spreadsheet. For the method comparison study, which I worked on for the remaining 2 weeks, For the method comparison study, which I worked on for the remaining 2 weeks, I were to compare the EPOC results to the reference instruments that was used in the hospital. So from my project protocol, 20 surplus lipid heparin samples were to be collected, and the panel of blood gases, electrolytes, and metabolites were ran onto EPOC. Right after testing the sample on EPOC, or in real time, the samples were then to be ran on the Siemens Rapid Point 500. Right afterwards, a portion of that sample were to be spun down and to have the plasma tested for creatinine and lactate on the architect. With the remaining sample, I would have to perform a spun hematocrit, as well as obtaining hematocrit from the Sysmex as well. And again, all the results were to be collected and transferred to Excel spreadsheet. So before we get to the results, I just want to quickly talk about the similarities and differences between the EPOC and reference instrument. It's definitely something to note considering we are comparing the results between the two, or between each instrument. It's interesting to note that the difference in technology between EPOC, Rapid Point, and Architect. Architect uses plasma, whereas EPOC and Rapid Point uses whole blood to test their samples. EPOC and Rapid Point both use potentiometry and ampermetry to measure their parameters, whereas Architect measures creatinine and lactate by measuring the change in rate of absorbance. When you're looking in the hematocrit, both 3 methods, the EPOC spun hematocrit, it says uses whole blood. However, they each have different methods of measurements. So EPOC measures hematocrit by AC conductivity, the principle being that the hematocrit is inversely proportional to the conductivity produced by the sample after it is corrected by the conductivity of sodium levels in the blood. For the spun hematocrit, it's basically blood that has been spun down or centrifuged, and then the hematocrit is read manually by marking each point within the glass capillary, separating the packed blood cells to the buffy coat and the plasma. And for Sysmex, hematocrit is measured via red blood cell pulse height detection method. So, while completing my project, there were some changes made to the initial protocol. For one, I were to include hemoglobin as part of my data collection. It's not part of my validation project, but was for an additional project that was happening at the same time. This second project focuses on the differences in hemoglobin results between various analyzers, that being Rapid Point, Sysmex, and EPOC. I should also note that EPOC doesn't actually measure hemoglobin, but offers calculated hemoglobin instead. However, the calculated hemoglobin from EPOC can help enrich the data for that second project. So as the result of the additional project, the initial target sample, which I mentioned before was 20, was changed to 30 to increase the N value. And now we'll talk about some of the challenges I encountered while I was working on my project. So one of the challenges I had while working on this project was finding samples with enough volume to complete all of the tests on all of the reference instruments. A couple of times I would be just short when I just need to run it on the last test, such as the spun hematocrit or the Sysmex for the hematocrit and hemoglobin. Or in another case, I would not have enough to repeat a test in a case of a QC failure on the test card for the EPOC. To make up for the missing test due to non-sufficient quantity, I had to collect additional samples to ensure that the sample size with all the complete tests maintained at 30. Getting samples with a wide range of results was also a challenge for me. I learned that we do need samples with ranges from one end to another in order to measure the instrument's ability to measure low and high values as to challenge the linearity of the instrument. To remedy this, I was able to collect surplus samples from MedSurg ICU, where the patient population tends to have extreme values in results. However, in other cases, such as finding samples with high hematocrit, that was really hard to find. In that case, I had to be able to sort of cooked up a high hematocrit from some of the samples. That means that I had to remove some of the plasma from the blood sample ahead of time, and then using the alterated blood with the high hematocrit and running that blood sample onto all the reference instruments as well as the EPOC. And that is a way to ensure that results from a wide range of analysis is collected. So another challenge for me was trying to minimize as much pre-analytical errors on my part as I can to ensure that the results that are measured are due to the instrument, hence the analytical part, and not due to any pre-analytical errors that can affect the results. Blood gases and lactate are particularly sensitive. For example, once the sample or QC material is exposed to air, partial pressure of oxygen can change, and it is important to minimize the exposure to air as much as possible and keep the samples capped. As well, we also have to measure the samples in real time. This is important concerning when we're measuring from EPOC to the rapid point. You don't want a huge time gap between those two, as the composition of the blood gases and even lactate can change. And I also have this issue with lactate specifically, just because from running the sample on the EPOC and then running that same sample onto the rapid point, and then having the sample spun down, there is a huger time gap between getting the sample spun down and then running that sample onto the architect, which can affect the lactate level, as lactate tends to increase as time increases as well. So that's something I have to be mindful of when I was working on my project and collecting the results. And this just goes to with time management and being as fast as you can. However, in the end, I was able to collect all the results for the 3 parts of my validation project and enter it onto Excel. For both the linearity and the precision study, the samples were to be collected, the results are collected and the values for each levels are compared to the acceptable ranges as provided by the system value assignment datasheet. This datasheet is lot number and sensor configuration specific and can be found on the EPOC website. For the linearity study, as I mentioned before, we want to verify the linear relationship between what's in the verification material and what the instrument actually measures, as well as ensuring its ability to measure high and low values. all of the parameters, the pH, partial pressure of oxygen and carbon dioxide, sodium, potassium, chloride, ionized calcium, lactate, creatinine, and hematocrit all fall within acceptable range as provided by the system value assignment data sheet. And I just have provided an example of potassium with one of the parameters, just because there are like 10 parameters. I can't show all of them in the presentation. And as you can see here for the calibration verification level, there are 5 levels, and I had to run each level in duplicates. And this is the acceptable range for each level. Similarly to the precision study, we are testing the reliability and the reproducibility of the instrument. And again, all of the values are compared to ensure that they fall within acceptable range. In this case here for potassium, with the QC material, there's 3 levels. And because I run them each in duplicate for 5 days, there's a total of 10 runs in total. And you can see here, this is the results and the acceptable ranges. So for my method comparison study, I ended up collecting 33 samples in total. The additional 3 was to make up for the NSQ samples. I want to mention that I did not participate in the actual analysis of my results, but from the protocol, the results were to be evaluated using linear regression analysis via EP evaluator program. I do have examples of analyses I've done on Excel, and it is shown on the next slide. And it's just to illustrate what kind of results we have obtained. Ideally, we will want a correlation coefficient from the linear regression analysis greater than 0.5, and the slope from the linear regression between 0.95 and 1.05. As you can see from the scatter graph for the partial pressure of oxygen, comparing EPOC and rapid point 500, we can see that the points or the results are very close to the line of the best fit. And you can also see that the correlation coefficient is at 0.99. This is compared to potassium, where those points are much more scattered around the line of best fit, with the correlation coefficient at 0.96. If you were to look at the same results using the Bland-Altman plot, where the y-axis is the y-axis is the difference between EPOC and rapid point, and the x-axis is the average difference between the 2 analyzers. You can see that for partial pressure of oxygen, the differences between the 2 analyzers are closer to 0, whereas compared to potassium, the differences are much more scattered away from 0. And I know there are more in-depth analysis for each parameter, and we also have to consider that each parameter have their own range of acceptable errors. But I just want to illustrate what kind of results I've obtained for each parameters. And some parameters, we would have a much more ideal result, whereas another parameter, it's not as ideal as we would have liked. But again, like more in-depth analysis was to be complete. So another part of validating for a point-of-care testing instrument that we have to consider is the operator's perspective. So the method evaluation only focus on the analytical part of laboratory testing. However, in point-of-care testing, we have to consider that other healthcare providers who are not laboratory focused will be operating these devices. And thus, we have to consider what potential pre-analytical errors that can occur when they operate these devices, as well as kind of determining its usability and how easy the instrument is to use. Pre-analytical errors include delayed testing for certain parameters. I know from the EPOC manual, they recommend that if you were to test for lactate, that it should be tested within 5 minutes. Or if you were to test for hematocrit, it should be tested immediately after collection to avoid blood from settling. Other pre-analytical errors include samples not collected in the proper tube, sodium or liftering heparins should be used on the EPOC. as well as introducing air bubbles into the sample, thus affecting the blood gases. However, I do feel like with proper training, these errors can be properly avoided. Overall, I feel like the EPOC is portable, intuitive, and easy to operate. The fact that internal calibration and QC checks are done within each card can prevent erroneous results from being reported, and this goes back to what I mentioned before about the operator's perspective, and that you can almost ensure that for the most part the wrong results won't be analyzed and used when it is invalid. As well, I do find that EPOC can be greatly beneficial to patient care, as it can provide both chemistry and laboratory tests, allowing doctors to make immediate healthcare decisions. I am also happy to hear, since I've completed this project in late December to January, that both EPOC has been validated, and so 2 of the EPOC instruments have been validated and released to be used in the operating room since early March. And to conclude, I would also like to add that I found this enrichment rotation to be a valuable experience for me. Not only did I get the opportunity to see the process of validating an instrument, I was also able to be involved within 2 projects, one being the validation project and the second with the hemoglobin project. I also had the opportunity to see and learn more about the point of care team and how they work with other healthcare providers to ensure that the point of care testing instruments can provide safe and reliable results. So thank you all for attending.
Okay, I guess I'll start then. As Laura introduced, my name is Sidney. I'll talk about the mass spectrometry enrichment rotation that I participated in when I was a student and doing my placement here. My sponsor was Dr. Andrea, and I want to give a special thanks to Dr. Bethany for showing me a lot of the process and a lot of the tests. So just as a quick overview, I'll go over why I chose mass spectrometry. I'll go over the different principles of mass spectrometry, the different separation techniques, the test performed at Toronto General, and my overall experience in the lab. So I chose mass spectrometry because when I was an undergraduate, everyone would always talk about how powerful mass spectrometry was and how there's a lot of applications for it. But I never really kind of connected it. That's what all the professors would say. So when I heard that I could actually participate in the mass spec lab, I really wanted to learn more about and how powerful it actually was. So mass spectrometry. So to discuss about it first, it's an analytical technique where molecules are ionized and separated based on their mass to charge ratio. So through this technique, you can virtually detect any molecule compared to the other analyzers, where they're kind of constrained by direct or indirect methods of measurement, or colorimetric methods or turbidimetric methods. It can also be multiplexed. So what that means is, for some analyzers, when you have one run, you can really only detect that one analyte that you're looking for. For mass spectrometry, you can have one run, and you can actually detect multiple charged species. So that's another advantage it has over other analyzers. It can also analyze small amounts of an analyte from the nanomole to femtomole region, depending on the technique you use. And just to reiterate again, a very powerful technique with many applications in pharmaceuticals research and, as we'll soon learn, in a clinical setting. So the mass spectrometer itself consists of 3 main parts: the ion source, the mass analyzer, and the detector. So I'll explain more as I show you some pictures. So the ion source is responsible for ionizing the analyte. And there's different ways of doing it. There's one where you can directly add a charge to it, or you can fragment it and it'll be charged that way. The mass analyzer selects for the charged molecule that you want. And through this, you'll see that there are 4 metal rods. Just as an example, you can actually change the radio frequency and the voltage to change the electromagnetic field. So you can select what charge species you want. And the detector, which is to detect the molecule pretty much, and it enhances the signal. You can actually see it as an output. And of course, you can't just put samples into your mass spec. There are a lot of constituents to your plasma, and you want to make sure that you purify your sample. So you want to separate it out from the other constituents. So there's many ways to do that. You can precipitate out your proteins that you don't want. It might be hard to see, but there's a small pellet down here that's the protein that's been precipitated out. You can also do liquid-liquid extraction, so based on different densities or hydrophilic or hydrophobic interactions with the molecule, you can separate it based on that. And you can also use other instruments as well. A gas chromatographer or a liquid chromatographer that will further purify your sample, and you can hook that up to a mass spectrometer, and that'll purify the sample even further before you put it into the mass spectrometer. So the 4 tests performed in the lab at Toronto General are methylmalonic acid, 11-deoxycortisol, fatty acid methyl esters, and amyloid subtyping. So for methylmalonic acid, it's a quantitation of MMA to monitor acquired or inherited methylmalonic acidemia. So what happens is, if your body lacks one of these enzymes to convert one substance into another, there's a buildup of this acid, and that causes acidemia in the body, and you can actually measure that. And it can be acquired or through diet or just autosomal. And this one is prepared through protein precipitation, and it's run through liquid chromatography and tandem mass spec. So you can see the liquid chromatographer here and the mass spec itself. And what you see here is the chromatograph of the liquid chromatographer. So what happens is the sample will go through and it'll come out at a certain time based on certain properties. And this peak here is the time that should be when the analyte actually elutes out, that comes out. And you want to make sure that you integrate it properly so that you calculate the right amount of area so that you don't integrate the wrong peaks as well. You can see that there's a lot of, I guess, noise you can call it, in this jaggedness, so you want to make sure you integrate it properly. The next test is 11-deoxycortisol. This one is another quantitative test. It's to measure the precursor, which is the precursor to cortisol, which is 11D, to diagnose and monitor 11-beta hydroxylase deficiency. So again, an enzyme is responsible for converting 11-deoxycortisol into cortisol, and you can monitor different disease states, such as Cushing disease. And again, for this one, it goes through liquid-liquid extraction, and it's also run through liquid chromatography and tandem mass spec. But when you look at the chromatograph here, you'll notice it's actually, the peaks are a little bit smoother, and there's a lot less noise. That has, compared to MMA, I'll go back, you kind of see it's more jagged here, smoother here. That has to deal with how the sample is prepared. If you put in more time and more steps to the sample, you'll get a cleaner run. But of course, there's other things to balance: time, cost, things like that. The third test is for fatty acid methyl esters. This one's a little bit different in that it doesn't actually use the mass spectrometer. It's a semi-quantitative test of your fatty acids in your blood, and you can use that to determine your risk of cardiovascular disease. So I'm sure everyone's ever heard of, if you want to be healthy, you want to take more omega-3s and less omega-6s. But now there's research that shows you want a certain ratio of both. You want a balance of both, not just all of omega-3s and no omega-6s. And this one actually just goes through a gas chromatographer, and you, based on the ratios of each, I think they measure 25 fatty acids, and you can measure that. And you can see in the chromatograph here, those are the different ones. But I'll zoom in one portion. Sometimes the software isn't perfect, and you can see that you'll see a peak here, and then this kind of little tiny peak attached to it. Sometimes the software isn't perfect, so the technologist will actually have to go in and make sure that you get the right base to integrate, so you calculate the right areas. It's another important thing. And the last test they do, and I think the most interesting test they do, is amyloid subtyping. So just to preface a little, amyloids are actually misfolded proteins. And so when a lot of these misfolded proteins are deposited in your tissues, if there's too much, your organs will essentially become non-functioning, and that can lead to organ failure. So at Toronto General, it's actually the largest referral lab for this kind of testing, and it actually takes a shotgun proteomics-based approach. So to break that down, what happens is your amyloid positive tissue is extracted. Once you extract it and prepare it, you run through liquid chromatography and tandem mass spec. And from that data, from the signal, you compare it to a database of known human proteins. And then based on how much coverage there is, you can identify the protein. And as of right now, there's over 35 subtypes responsible for many disease states. I think when they started it, or when they first conceived it in 2009, or 2010, there were maybe only 12. And there's a lot of research that shows that a lot of these amyloids cause a specific disease state. So it's not an association of a disease state. It actually causes the amyloidosis state. And the treatments can vary. For example, if you found one of the amyloids to be transthyretin, has a lot of cardiac issues in the body, or causes a lot of cardiac issues in the body, and you would actually need a liver transplant for that. But if you found a different amyloid to be a light chain, for example, either kappa or lambda, you would need stem cell transplant and high dose chemotherapy. So just by talking about it a little bit, you can see how specific the technique is and how powerful it can be. So to go over the process just a little bit more, it starts off in histology, so suspected tissue is actually stained with Congo red, and this is still a gold standard in detecting the amyloids within your tissue. From there, it actually undergoes laser microdissection. So a laser will actually cut out the portions of positive tissue, and it'll be collected in the microtubule, and then it's treated with heat and other solutions to make the amyloid as linear as possible, because it's all misfolded. You want to make it as linear as possible. From there, you treat it with trypsin, so it's cut into smaller peptides, and you run that through the mass spectrometer. From that signal, you can actually look at the protein identification report, and based on the unique signals, how much of the signal, and how much coverage there is, you can determine what subtype it actually is. So to show some pictures, so at the bottom is actually positive for Congo red staining. So you can actually see the amyloid plaques here, these red portions. And so the pathologist will actually put it in this microscope and laser microdissector combo. He'll put it in there, and he'll actually on his pad draw out the positive portions for the laser to cut out. And so you can see like a before and after picture here. And here in this little GIF, you can actually see the laser cut procedurally along like the circle or square or whatever shape he cuts. From there, after it's collected, it's treated with different buffers and different solutions, and also heat to make it linear. And then it's cut with trypsin to make it into smaller peptides, and it's run through the mass spec here. This is the Orbitrap that they have down, and it's one of the newer mass specs with a different technique to it. It gives a higher resolution, so it's more powerful, if it wasn't already powerful enough. And from there, you compare it to your database, and you can determine what protein it actually is. So at the bottom is actually like a small cleaned up version. So the green are actually just controls to make sure that everything is running okay and the identification is going okay. But right here on the, if I can get my mouse, the fourth line here, you can see it actually says transthyretin with a coverage of 64%. So that would most likely be the identification of the amyloid. But the pathologist and the biochemist will actually, on their own, come to their own conclusions and then come together and make sure that they have the same conclusion. And if it is, then the identification is most likely right. Just a few statistics to show. When they started doing this test in, I think, April of 2015, it was around 50, and since then it's been steadily increasing. It doubled within the next year, and it's still increasing to this day. The source of the samples, they vary, but a lot of them come from within Ontario. They can come from any of the hospitals or private labs, like Sunnybrook or Dynacare, but it can also come out of the province. So you can have it from Vancouver or PEI even. And a lot of the amyloids identified are, almost 3/4 are actually light chain, lambda or kappa. The next biggest chunk would be transthyretin in the red, and then some of the other small ones are just account for a small percentage of it. I think serum protein A is just another big chunk of it. So in summary, I definitely learned a lot during my mass spec rotation. Of course, I got to observe a lot, and I got to actually be engaged in a lot of the pipetting and integrating. But I definitely learned more about mass spec and how it fits and how actually powerful it is. Now I understand what that means in a clinical setting. And it made me appreciate and see a bigger picture of how the hospital operates, especially in diagnosing, because as an MLT, you only, I guess, really see the middle portion when you kind of do all the testing. But with this, you can appreciate, from a little bit of the beginning and a lot of the testing to the end for like diagnosing and treatment and identification. You can definitely see how it fits together in kind of the hospital setting. So I definitely really like learning about that. Just a few references and just a few more acknowledgements. I want to thank Dr. Andrea for sponsoring me, and Dr. Vathany for showing me a lot of the tests, the technologists for actually allowing me to shadow them, and Peter Bridge for allowing me to actually partake in the enhancement rotation.
All right, so before I let you sit down, does anybody actually have any questions for either Sidney or Thuy? Yes, Joe?
We had that your samples needed to be either sodium or lithium, but does the EPOC measure just potassium? Or, because if you're measuring all the electrolytes, then you can't have sodium.
No, you can't. If you're measuring all this electrolytes, you can't have use sodium lithium heparin. But like you can also to measure creatinine. You don't have to measure electrolytes. You can select which test you want.
So that means then if you end up collecting it in a sodium pepper tube.
Then the electrolytes it will be correct.
Yeah. Okay, because I just saw that and I was perplexed by that.
That's why there's an option of sodium or lithium heparin.
All right, so thank you, Sidney and Thuy, for presenting today. We actually have gifts for you for presenting. So here we go. And if you guys can select your winners for the there's two, so each of them pick one. I don't know if you want to do it in one night
Keep this coupon.
Ticket 635259.
Okay. And the next ticket is 635289. Hey! Those are one.
Great job, guys.
Oh, thank you.
All right, so thanks again for coming. Hopefully you all enjoyed the lectures that we had this week, and make it for the lunch tomorrow, 11:30 till 1:30.